Yeah, usually at the end of a couple of day meetings, I'm a little bit worn out, but here I'm really thankful for bringing us together and just being part of that. So I want to talk about cell division asymmetry, which is also the form of stochastic variation, which I will show you or illustrate a little bit, and then see how sensitivities to that or deviations from that are so if we think about cell division, it's basically one of the roots of life. So without cell division, there would be no life, and without asymmetry, there would also be no life. So if everything would be symmetric, you could think this is kind of the way of thinking, or the simplistic way that predominates, at least in the bacteria, for a long time, that they just divide symmetrically, and that provides a way of that the population projects. But in fact, what we see is there's accumulation of damage, and so if there would be perfect symmetry with increasing age, basically the population would accumulate more and the damage and one point splash. So one of the nice solutions is that if we have a little bit of asymmetry in this division process, we get heterogeneity, and we basically get a robust and stable system. So this asymmetry is then also kind of the foundation of division of labor that leads to evolution of multicellularity. It explains the evolution of aging. So for that, I think it's a valid point of thinking about it. How we quantify the asymmetry is usually using the mother machine. So it's I want to just start the video. Oh, no, video doesn't. Okay, so the video doesn't work, sorry. So this is the mother machine. So it's we heard about it yesterday. So it's a PDMS chip on a slide, and so here's inflow and outflow. There's thousands of these small side channels where we have really high control, so stability control, the media is controlled, and in these side channels you get an end of the media at the 0.2 seconds. So it's always a question, is there still variation? So when we publish that, they all ask, is there variation in these channels? So what you see here is the side channels. So the bacteria sit here, they grow and divide. This is what we call the mother cell up here. This is a phase contrast view, and this is the fluorescent imaging view of a transcription factor or optimizer. Fluorescent promoter in an operon. And you see there is variation between the different mother cell lineages, but also within the cell lineages. We look into that. So here is the timeline, and we have here each line is one cell. So we see different, the lighter green, the higher the transcription factor signal. So we see there is basically periods where they go in more transcription factor, and the transcription factor I'm talking about is RPRS, which is its general stress response. It regulates about 10% of all equal actions. It also has some link to fitness or growth and division. So if you have upregulation of fluorescence, so in RPRS, you basically have grow slowly, and growth rate is very closely related to. And Audrey is the postdoc that did the data collection on that, so I won't mentioned her as well. So one of the questions is now, how does such stochasticity influence fitness? And the one question is, when we see, okay, the cell is growing and dividing, so what is the optimal asymmetry, or is there no asymmetry when dividing? And the other question is, if there is such an optimal asymmetry, how sensitive is it, this optimum? So if we perturb this optimum, how much fitness do we get? And if we look into here the daughter cell and the mother cell, we see that there is actually an asymmetry in the length. So previously people thought, well, they are symmetrically dividing, morphologically symmetrically dividing. That actually is not true for E. coli at least. And also for transcription signal, we see the reverse, that the mother cell has slightly lower transcription signal than the daughter cells. To answer this first question here, what we need is the link between the difference in the or the difference in asymmetry to fitness. And for the second question, we basically look into the variance division asymmetry and try to do that. The link to fitness, what we have is this single cell growth data on cell growth and transcription factor dynamics. So what we want to have is how to link this to population growth, which I defined as fitness. And for that, we use structured population models. So it's just a matrix model where you have a transition matrix and a population vector that you multiply with this transition matrix. So imagine you have juveniles, non-reproductive adults, and reproductive adults. You just look how many of these you have. You have probabilities at each of these different stages that they survive, that they transition to another state, and that they reproduce. And that way, you basically just project it forward, and that gives you basically population. This is what we do for the system here, except that we not just have a few states, we have more states. So we have length, and we have the transcription factor signal, we have the survival, so highest survival at smaller cells with higher transcription factor signal a little bit. Division probabilities, only larger cells with lower transcription factor signal divide. This is also understood because we know that transcription factor signal and growth rates are negatively correlated. So these cells are just not growing very fast. And then we just multiply that, and that's basically like one of our first entry for the model. You see a little bit of a trade-off between survival and division as well. And in addition, what we see is that, okay, the cell can grow and not divide. And then, so it changes its size and it changes its transcription factor signal. At the same time, when the mother is dividing, so then we can measure the size of the mother from this point before it divided to the point when it divided, and also the transcription factor signal. And we can do the same thing, just at division, what is the size and the transcription factor signal of the daughter. And here, this is now for mother-daughter transcription probability. So if you have a mother of that size with a RPOS level of that, this is the type of daughters that this mother is producing. So the daughter length and the RPOS of the daughter. So that's kind of what we use. Then we plug this in and get our growth rates. And this is the observed growth rate from just putting in our data that we collected. What we then can do is we can change the asymmetry, division asymmetry. And when we reduce the transcription factor and the size asymmetry, we see this is the observed asymmetry here. So basically what we do is we reduce this difference between these two, then we get a reduction in the cross rates. We can do the same thing by increasing the asymmetry. So we make these more different. And again, we get a reduction in the growth. So at the currently observed asymmetry here, we get this optimum in the growth rate. And if we deviate from that, we basically get no growth rates. So now what we can do is we can change kind of the noise, the variance in growth, in division asymmetry, in growth between, or the size between the mothers to mother and mother to daughter when they divide. And this is simply like that. We just basically have our transition probabilities. So we just increase the variance in these probabilities. Here the variance, we increase the variance in length, or the variance in transcription factor signal, or the covariance between the two. And if we do that, we see that this is the mother-daughter transcription factor variance. Then we see basically lower variance gives us higher population growth. This is a little bit expected, but if we, for example, look for the mother-daughter size variance, then there's basically no impact of that. And if we look into the other components, so the covariance or the variance or stochasticity in growth or transition between transcription factor signal from time point 1 to time point 2, there's no effect on fitness. There's no effect on the population growth. So that would suggest that cell intrinsic variance in growth and gene regulation, generalizing here, and growth asymmetry is under weak selection. So the only thing that is somewhat under selection in terms of variance remains this asymmetry, the optimal asymmetry in the mother-daughter transcription factor variant. So yes, to some degree, division asymmetry has evolved an optimum. And how sensitive is that? So selective forces drive cell-to-cell heterogeneity. As heterogeneity rises through division asymmetries, and we see this cell intrinsic transcriptional noise, but it still has consequences. And then on the other hand, cell-to-cell heterogeneity can have clear consequences in terms of cancer or phenotypic antibiotic resistance, but also helps for regeneration of stem cells, for example. So with that, I'd like to end and thank members of my group and some collaborators and funding studies.